Welcome to the world of health and disease. Now in this session we are going to discuss the next part of the deadly disease, contagious disease, AIDS. Now with respect to the disease AIDS, as it is a contagious disease which is easily spread from one person to another person. Now what is the role of this virus, HIV virus, where we also call it as a human immunodeficiency virus, where we also call this as retrovirus. HIV virus is also called as retrovirus. Why it is called as retrovirus? This is because it is having the ability of converting its RNA into DNA. That means it undergoes the process of a reverse transcription. Okay. So as you have studied the molecular basis of inheritance, so in the molecular basis of inheritance, RNA, if it is converted into DNA, we call the process as a reverse transcription. But generally from the DNA, mRNA will be synthesizing, where we call it as transcription. But here it is a reverse, from RNA, DNA is produced. As a result, with the help of this process, reverse transcription by an enzyme, reverse transcriptase, this HIV virus is also called as a retrovirus. That means which can undergo reverse transcription. So the life cycle of this retrovirus we are going to study, how exactly it replicates, it multiplies in the host cell, depleting our immune system, and so it makes the immune system very weak. That is why AIDS is said to be acquired immunodeficiency syndrome, as the immune system gets a deficit. Okay. So here we are across the life cycle or the replication of the retrovirus, which is most important. This is the schematic representation which will be asked separately for a fixed 5-mark level question. Remember, okay. So most of the times this diagrammatic representation, it is not a diagrammatic, it is a schematic representation, because we are writing in words. Diagram is different, schematic is different. This is the schematic representation of the replication of the retrovirus in the host cell. Either it is a helper T cell, either it is a macrophage cell. Okay, so in those cells, how exactly the retrovirus gets multiplied. Okay. So let us start with the first one, retrovirus. When the retrovirus takes the entry into the human body, then what exactly it will do? Okay. Now if we look at the retrovirus, this retrovirus is having a viral RNA core. So outer part is the viral RNA core, and sorry, not outer part, inner one is the viral RNA, it's made up of RNA. Then outer part is the protein coat. Outer part protein coat, inside it is having viral RNA. Okay, so core is there. So core is the central part of this virus. When this virus, retrovirus, it infects the normal cell. This is the normal cell, where we call it as a animal cell. Okay, it is going to infect the animal cell. So let us take this as a macrophage cell or a helper T cell. Okay, because helper T cell, as we have discussed, it is the most strongest cell and their number is also abundant. So these helper T cells are infected by this virus. So virus infects normal cell. Then when it infects, as you know, this virus, in the previous year you have studied this virus, as it is having its genetic material here in the form of core, RNA core, in the protein coat. So it will inject through this capsule and a sheath. Okay, so there is a collar like structure here, then elongated sheath is there. Here these are the tail fibers, it attaches on the surface of the animal cell, then it will inject only its what? Genetic material, RNA. Okay, the entire protein coat is not injected into the cell, only its RNA core is injected. So that is why what we say, viral RNA introduced into the cell, normal cell. Okay, now the process we can call it as infection, because the viral RNA has entered into the cell, and this is the cytoplasm of the cell. So we can call this as the cytoplasm of the cell. and outer part of the cell, as you know it is made up of cell membrane, where we also call it as plasma membrane. Okay. So, this is the cytoplasm of the cell. Inside the cytoplasm of the cell, viral RNA is injected. Okay. Now, this viral RNA is single stranded, as you know it, DNA is double stranded. Okay. Now, this RNA is converted into DNA with the help of the enzyme that is produced by this viral RNA only, and the name of the enzyme is a reverse transcriptase. This is an objective question. Which enzyme is responsible for conversion of RNA into DNA? It is a reverse transcriptase. The process of a conversion of RNA into DNA is called as a reverse transcription, and the enzyme is a reverse transcriptase. Followed? Okay, so that is the basic difference. Okay. Then from RNA, DNA is formed. This DNA is present in the cytoplasm. Now, this DNA gets incorporated, inserted, entered into the nucleus of the host cell. That is why we have written here viral DNA. Look at this. This is the DNA. Viral DNA incorporates, gets inserted, enters into host genome. Genome is nothing but it is our genetic material. Okay. So, it gets entered into the nucleus. Inside the nucleus we have our genetic material DNA, and it gets attached to a part of the DNA. Then after that, what will happen? From this, it makes our host cell as its factory, virus factory, and this DNA starts to produce a RNA. Which RNA? Viral RNA. Along with our RNA, that means other part of our host DNA will produce normally our RNA. Along with that, this viral DNA starts to produce viral RNA. So, that is why new viral RNA, look at this, new viral RNA is produced by the infected cell. The cell has already infected. Now, our helper T cell, instead of killing the antigen, it starts to obey the rules of the viral DNA. Okay. And this DNA, it becomes the master copy, where it starts to produce a lot many number of viruses. Okay. So, here viral RNA is produced. Now, this viral RNA inside the cytoplasm of the cell, when it is released, it will develop the protein coat. Protein coat is produced. So, that is why new viruses are produced in the host cell. Later, these new viruses, either they are released one after the other, or there is a rapid multiplication. When the virus number becomes huge, they may burst the host cell and they are released out. Okay. And this new virus which is released, it infects the neighboring cells, and the process goes on continuing. Again it is released, it comes here, it infects one more helper T cell, same process is carried out, then again released, again go and infect other neighboring cell. Look at this, it is going to destroy, deplete our own host cells. This is why our immune system goes on becoming less deficit. That is why AIDS is said to be acquired immunodeficit syndrome. Followed? So, this is the life cycle of a retrovirus, which is most important, and it will be asked for a 5-mark level question. Okay. So, we are stressing more on the life cycle of a retrovirus, where we also call it as replication of the retrovirus in a host cell. Either it is helper T cell or either it is a macrophage cell. Okay, where the viral RNA gets incorporated, forms a DNA, DNA gets incorporated into the host genome, and from the viral DNA most of the RNAs are produced, viral RNAs are produced. Okay. And after the release of these RNA, they are going to infect the neighboring cells. Okay. So, this is how the life cycle continues. Okay. So, this completes the replication of the retrovirus, and here we are ending up with the contaminated disease AIDS. And with this we are moving on to the next disease. The next disease is cancer. Okay. Now as we have came across the basic difference between these two different types of deadly disease, but out of these one is infectious, that is easily spread from one person to another person. So that we have discussed till now. And next one is a non-contagious disease, that means it is not easily spread from one person to another person, and it will be confined in that person who has got infected with the disease, and the disease name is cancer. So let's look at the next disease, cancer. Now as I told you, cancer is a non-contagious disease. It is not spread from one person to another person. Suppose if a person gets infected with the cancer, it might be harmful for him or it might be deadly for him. That means none of the diseases are good for health. That means, okay. So suppose in the classification of the disease, ಯಾವ disease better ಅಂತ ಕೇಳಿದಾಗ ಕೆಮ್ಮು ಚಲೋನೋ ನೆಗಡಿ ಚಲೋನೋ ಯಾವುದು ಚಲೋ ಅಲ್ಲ. None of the diseases are good for our body. Ultimately we are going to suffer. Either we get a simple common cold or a cough, pneumonia, okay, elephantiasis, whichever it might be. None of the diseases are good for health. ಯಾವುದು ಚಲೋಪ disease ಅಂತಂತಂದ್ರೆ ಯಾವುದು ಚಲೋ ಅಲ್ಲ. Okay, we should be healthy, we should be cautious about our health only. Understood? Okay, so that's why this cancer, it is going to affect the humans either to a lesser extent or to the maximum extent. Maximum extent here it is death only. Okay. So what exactly is cancer? Cancer is nothing but it is a uncontrolled mitotic division of the cell. So cancer, it can be defined as it is uncontrolled mitotic division of the cell. Uncontrolled mitotic division of the cell is said to be cancer. Now here there is an uncontrolled way of cell division where the cell division does not stop. As a result, the cell number goes on increasing, goes on proliferating, leading to a mass of cell where we call it as tumor. Okay. And with respect to the cancer, almost in India only, there are more than 1 million people who are suffering with the cancer, and out of which many have died. Okay, major cases, death is there, right? So almost remember the number. So how many people with respect to the AIDS? 25 million people all over the world, that is all over the world. This is more than 1 million people are suffering with the cancer in our country, in India. The previous disease was in the world, this is in the India only. Now also we have cancer patients, we have big cancer hospitals also, okay, where the treatment has been given, but some can be cured, but majority of these they cannot be cured. Only the thing is we are prolonging the death period. We are prolonging the death period, and it depends on the severity and type of cancer, because in the cancer we have different types of cancers. Lung cancer is there, oral cancer is there, cervical cancer is there, uterine cancer is there, ovarian cancers are there, prostate cancers are there, right? So different types of cancers are there, okay, different based on different agents, okay. Everything, most of that, it depends on our lifestyle, food style, all these things, okay. So everything will also count. Along with that, one more hidden term is there. Why do we get cancer? I will tell you. Okay, what is that? Okay, when we go through the entire part of the cancer. Okay. Then cancer causing agents. Let me take that term. Cancer causing agents, we call them as carcinogens. Okay. So before going to the causative agents, let us look regarding the types of the tumors. Now, Masses of cells, group of cells, uncontrolled division leads to proliferation of the cells, where this proliferation of the cells leads to the formation of the tumors. So, proliferation of the cells leads to formation of tumors. That means tumor is group of cancerous cells. It is called as a tumor. These tumors are of two types. So, there are two types of tumors, namely the first one is benign tumor, it is a localized tumor. Benign tumor is localized tumor, and second type of tumor that we are going to come across is malignant tumor. So, it is non-localized. So, which are the two different types of tumors? Benign tumor, malignant tumor. Benign tumor, that means when the cells start to multiply rapidly, they form into group or masses of the cells where there might be an bulging portion also in any part of the body. Okay, it swells in that area. So, we call that type of tumor as benign tumor. That benign tumor is localized. Localized meaning it is present at a very particular part of the body or any particular part of the body, which causes less harm. Which causes less harm, because this is located at only one location of the body. That is why the term localized. It is a localized. But look at the next part, which is more deadly, malignant tumor. This is non-localized. Non-localized means when cancerous cells starts to develop in our body, then from one part some of the cells gets slowed off. Slowed means removed. They migrate to other body parts, then they start to proliferate there, where we call it as neoplastic cells. Okay, new cells are cancer cells are formed in other part. Here it gets slowed off, it moves, again it goes to another body part, again it starts to proliferate, where we call it as neoplastic cells. Again it forms tumor. From here again it spreads to other body parts. Look at this. That means a malignant tumor, it is non-localized. It spreads from one part of the body to other body parts, where we call it as metastasis. Metastasis is the major property of the malignant tumor. It is an objective question again. What is metastasis? Okay, so let us continue this. That is the definition of the metastasis also. Okay, so malignant tumor, it is non-localized. So it moves or migrates from one part of the body to the other part. This property or this process, okay, so let us use the term, this process is said to be metastasis. Okay, so this is said to be metastasis. Then what is metastasis? Metastasis is nothing but the movement of cancerous cells from one part of the body to the other parts is said to be metastasis. As it is the continued part, I have written in this way. but when metastasis definition is asked, you have to write in this form only. The movement of cancerous cells from one part of the body to other body part is said to be metastasis. Followed? So, that is the definition. Now, why these cancer cells proliferate? This is because, let us put in a star mark, normal cells have the property of contact inhibition. Normal cells have the property of contact inhibition, while cancerous cells have lost this property. Remember what is contact inhibition. Normal body cells have the property of contact inhibition, while cancerous cells have lost the property of contact inhibition. What exactly it is contact inhibition? That means whenever the skin gets ruptured, the skin has to be reformed by the process of mitosis. We know this. That means our cells, if it is damaged, then ultimately through the mitosis it gets recovered. Then after that, when the entire process is completed, then one cell, when it comes in contact with the other cell, it inhibits the cell division. It stops the cell division because skin is reformed, then ultimately it stops the cell division. This is what is said to be contact inhibition. While cancerous cells, they don't have this property, they do not have this property of contact inhibition. Hence, the cells starts to multiply like rapidly like anything, in such a way that they start to compete with the neighboring cells for the purpose of the food, so that the other cells, neighboring cells should starve and they should die. When neighboring cells start to starve and die, cells will die, tissues will die, organ will die, organ system collapses, person will die. Okay. So, this is what is done by these cancerous cells. Okay. So, remember, normal cells have the property of the contact inhibition, while cancerous cells have lost this property, right? Okay. Now, what is the main cause for the cancer? Let us go for causes of cancer. So, let us come across a definition here: carcinogens. So, carcinogens means cancer causing agents. Cancer causing agents are said to be carcinogens. This is the definition. Why cancer is caused? Due to carcinogens. What are these carcinogens? Cancer causing agents. And these carcinogens might be different agents: physical agent, chemical agent, biological agents. So, there are three agents. So, let us look at the physical agents. So, ionizing radiations like X-rays, gamma rays and non-ionizing radiations like UV rays causes cancer. Ionizing means they have enough energy to break the bond between the DNA, and non-ionizing, they are having less energy to break the bond, but still they breaks the bond and it leads to mutations also. So, physical agent, ionizing radiations like X-rays, gamma rays. X-rays and gamma rays, that means if we are getting exposed to the X-rays or gamma rays, it is quite harmful because these are cancer causing agents. X-ray, it will change the structure of the DNA, there might be mutation, and that mutation may lead to what? Formation of the cancer. Gamma rays, quite harmful. UV rays, as you know, ultraviolet rays coming from the sun, it is trapped by the ozone layer. So, if ozone gets decreased, then ultimately if UV rays are emitted from the sun to the humans, then ultimately it is going to cause the skin cancer in the humans. Okay. So, hence these physical agents are responsible for cancer. Okay, this is one thing. Then second one is chemical agents. So, chemical agent is nicotine present in tobacco. Okay, so nicotine present in tobacco. As you know, tobacco contains an alkaloid called as nicotine. Nicotine is quite harmful, that leads to various different types of cancers. Okay, if the tobacco is smoked, then lung cancer. If tobacco is chewed, then oral cancer. Okay, so oral cancer. Then third one is biological agents. Biological agents like oncovirus. Okay, so onco refers to cancer, cancer causing virus. So, study of cancer we call it as oncology, and cancer doctors are called as oncologists. Study of cancer is oncology, study of cancer. Okay. So, here biological agent refers to virus. There are certain viruses called as oncoviruses where their number is very less. Otherwise, if this virus would have came to most of the people, ultimately it would have killed with the cancer disease only. Okay. So, biological agent, so it is through the oncovirus, cancer causing virus. Now, out of the three, the most deadly, do you know which is most deadly and most quite wide causing disease? It is chemical agent. It is chemical agent only. 99% of the cancer is due to the chemical agents. It is due to nicotine. It is due to tobacco. Either smoking or chewing, it is going to cause cancer definitely because of this. So, whenever a people or the people they intake either tobacco directly or in the form of gutkhas or smoking, so there will be a statutory warning that chewing or smoking up of the tobacco causes cancer. And nowadays they are giving the photography of the cancer also. Okay, oral cancer, throat cancer, lung cancer, everything. So, even with that statutory warning, if the person neglects and go for the addiction of this tobacco, then definitely he is going to end up with his life with cancer. Okay, because in the society we come across most of the people who tells that from 20 years, from 30 years I am chewing the tobacco, I am smoking, but nothing has happened to me, but they don't know what is going to happen in future. Okay, within few days or within few years what is going to happen, they are unaware of this. Okay, it's not a great thing, or if a person smokes, then it doesn't mean that he feels proud, okay, or bureaucrat or a aristocrat. Okay, it's not like that. Okay, he is unaware that he is going to get the cancer. Definitely 99.9% of the lung cancer is due to smoking, either it is active smoking or a passive smoking. So, irrespective of that, but for the smokers it is going to cause 99.9% of the lung cancer is by smoking, as you know it, which you have studied in the previous year, respiratory system. Okay, disorders of the respiratory system. So, chemical agent is one of the most deadly agent that we find it throughout the world wide where the people are getting cancer. Okay. So, physical agent their number is less, biological agent their number is less, vast number is chemical agent only. Okay. It is just like inviting cancer by ourselves if we are taking this such type of tobacco. Okay. So, irrespective of the forms. Then apart from this, there are certain genes in our body. There are certain genes in our body, that means in our cells there are certain genes. We call those genes as proto-oncogenes or cellular oncogenes. That means in the cells, proto meaning primitive or the first type of the genes which are suppressed, the genes are suppressed. In certain conditions, if the genes are triggered, then the person may get cancer, because we can find such type of person, if he has got the cancer, when he goes to the doctor after his symptoms and all those things, after diagnosis and detection, when doctor says you have a cancer, then their reply might be, I am not a drinker, I am not a smoker, I don't even chew the tobacco also, I don't have any of the bad habits, but why I have got the cancer? Then doctor will say, you have your own genes, that means cellular oncogenes. There are certain genes which are present in our cells, they are suppressed. We don't know under what conditions they get stimulated, under what conditions they get activated. If they get activated, then definitely it is going to cause cancer. And remember, all the colored foods that we are going to eat are carcinogens, cancer causing agents. All the colored not fruits, all the colored agents. Okay, that means whenever we go to the bakery, we have different colored cakes, and we don't know what chemical they have used, green color, pink color, red color, orange color, yellow color. Okay, now those chemicals, even in case of the fairs ಅಥವಾ ಜಾತ್ರೆ ಒಳಗಡೆ ಬೆಂಡು ಬೆತ್ತಾಸು ಇರ್ತಾವೆ, ಬೇರೆ ಬೇರೆ ಕಲರ್, ಪಿಂಕ್ ಕಲರ್, ಗ್ರೀನ್ ಕಲರ್, ಯೆಲ್ಲೋ ಕಲರ್, ಆರೆಂಜ್ ಕಲರ್. Okay, now what do you do in the birthday parties? ಅಲ್ಲೊಂದು ಮೇಲ್ಗಡೆ ಫ್ಲವರ್ ಇರುತ್ತೆ, ಪಿಂಕ್ ಕಲರ್, ಯೆಲ್ಲೋ ಕಲರ್, ರೆಡ್ ಕಲರ್. You will fight with your friends to eat that flower from the cake. It's a carcinogen. Think of it next time. Okay, and whenever you eat it, ಕೆಲವೊಂದು ಸರಿ ತಿಂದಾಗ ಏನಾಗ್ತದ, you find a little bit bitter taste in the tongue. Carcinogen. It's a carcinogen. ಹಂಗಿದ್ರೆ ನಾವು ಒಂದು ಸರಿ 2 ಸರಿ ತಿಂದರೆ ಬರ್ತೈತೇನ್ರಿ? ಬರಂಗಿಲ್ಲ. ಯಾವ ಬರ್ತೈತಿ ಹೇಳಕ್ಕೆ ಬರಂಗಿಲ್ಲ. Understood? So this is what we call it as an idiopathy condition, right? So that's why these chemicals may also trigger the genes in such a way that our inactivated form of the genes may get activated. In what conditions we can't predict also. Okay, so that's why everything it depends on the, that's why I told you, health aspects. That means our lifestyle and food habit will also decide what type of disease we are going to get. That means if you are having a good healthy food habit and a lifestyle means what? Walking, running, any type of games, playing games or exercise, everything ಇವೆಲ್ಲ ಇನ್ಕ್ಲೂಡ್ ಆಗ್ತಾವೆ ಇಲ್ಲಿ. ಇದಕ್ಕೆ ನಾವು ಲೈಫ್ ಸ್ಟೈಲ್ ಅಂತ ಕರಿತೀವಿ, ರೈಟ್? ಅವಾಯ್ಡಿಂಗ್ ಅನ್ನೆಸೆಸರಿ ಜಂಕ್ ಫುಡ್ಸ್. ಓಕೆ, ಆಲ್ ದೀಸ್ ಆರ್ ದ ಗುಡ್ ಲೈಫ್ ಸ್ಟೈಲ್ಸ್. ಓಕೆ, ಸೊ ದಟ್ಸ್ ವೈ ಲೈಫ್ ಸ್ಟೈಲ್ ಮೀನಿಂಗ್ ವಿತ್ ರೆಸ್ಪೆಕ್ಟ್ ಟು ದ ಹೆಲ್ತ್ ಈಸ್ ಡಿಫ್ರೆಂಟ್. Okay, lifestyle with respect to the health part is different. So we should know this, right? Okay, so this is regarding the causes of the cancer. Then cancer diagnosis and detection. So we are at the last part of the cancer. So cancer diagnosis as well as detection. So how it is diagnosed, how it is detected, and what is the treatment for this. And regarding the precautionary measures, as we know, whatever the causes are there, those causes has to be avoided. So X-ray has to be avoided, because if a person gets fracture, then he will go for a doctor for an X-ray. But for the X-ray, continuous taking more X-rays in a year is not good for health. Suppose if you are going across the X-ray once or twice, doesn't matter, but frequent X-rays are not good for health. And even the pregnant females, for them X-ray is not done, because those radiations may also affect on the developing fetus. Okay, so let's go to the detection and diagnosis. Okay, so here in the detection diagnosis we have the biopsy. So mainly the biopsy is done. So biopsy and histopathological studies. Okay, mainly for the detection of the disease. So, biopsy and histopathological studies of the tissues blood and bone. Okay, biopsy is nothing but, suppose if we suspect if there is a tumor, ಒಂದು ಗಂಟೆ ಆಗಿದೆ ಅಂತ ಅನ್ಕೊಂಡು, if it is a tumor, some part of the tissue is taken, then it is studied under the microscope regarding whether it is having the property of contact inhibition or no. Okay, so that is a biopsy. Part of a tissue is taken and it is diagnosed. So that is done by the pathologist only. Okay, so that histopathological studies of the tissues, if there is the blood cancer, even for the blood cancer, it is checked. If it is a bone cancer, part of a small part of a bone is taken and it is detected. Okay, so this is one thing. So where we can detect as well as diagnose. Then apart from this, especially in the blood, we have the condition called as a leukemia. Leukemia is nothing but it is a white blood cancer. Okay, where we generally say it is blood cancer, but it is a leukemia, white blood cancer only. In the blood cancer persons, the blood is detected. So leukemia patients, so their blood is detected. Then apart from that, there are other technologies like the techniques like radiography is used, use of X-rays, then CT, where we call it as computed tomography, then MRI, this is magnetic, resonance imaging. So especially X-ray is done for the hard tissue cancer, like the bone cancer, we can go for the X-ray. For the soft tissue, we can go with the CT scan as well as MRI. CT is computed tomography, MRI is magnetic resonance imaging. Okay, we can go with these two. Okay, for the internal organ cancers. Okay, now especially this CT scan, it uses X-rays to generate a 3-dimensional image of the internal organs. So here it uses the X-rays to identify the 3-dimensional images of internal organs. Okay, so this is CT scan. Then MRI, it uses strong radiations. Okay, mainly this MRI, it uses strong magnetic fields and non-ionizing radiations. To detect the cancer of the soft tissue. So we are implementing these two. So strong magnetic fields and non-ionizing radiations to detect the disease. Then apart from this, even antibodies are used against cancerous cells. Okay, that means the antibodies which can attack and identify the cancer cells, even though such type of antibodies can be used to detect the cancerous cells. That means if the antibody gets attached to these cells, definitely the cell is said to be cancer. So all these are the detective process, detection as well as diagnosis. Yes, after diagnosis, now suppose if we come to know that the person is suffering with the disease cancer, then what is the next process? We have to go with the treatment. So let's look at the last one, treatment. Now here better to avoid all the agents here, physical, chemical as well as biological agents from which the cancer is caused. Then our food habits. Okay, so any type of chemical if we consume, it might be quite harmful also. Even the chemicals that we are spraying in the agriculture field, that is also quite harmful. Okay, last one, treatment. For the treatment, what we use, what are the procedures that we are going to follow. So here we have the one is surgery, radiotherapy, immunotherapy, chemotherapy, and apart from that we have a modified interferon called as alpha interferons. And with this treatment also there are certain side effects like nausea, okay, then anemia, hair loss. Now here surgery in case of the benign tumor, if a tumor is localized, it can be cut and removed through surgery. Then radiotherapy, through the radiations cancer cells are killed. Immunotherapy or chemotherapy, here we are giving certain chemicals in the form of injections or in the form of tablets, so that the cancer cells can be controlled. Okay. Then apart from this, nowadays we are using modified interferons, where we call them as alpha interferons. When they are injected into the blood, they will target the cancer cells, where they can go and kill only the cancer cells apart from our normal cells. Okay. But with any kind of this treatment, either it is a chemotherapy, immunotherapy or radiotherapy, there are side effects. Those side effects includes nausea. Nausea means giddiness, ತಲೆ ತಿರುಗಿದಾಗ ಆಗೋದು, ಸುತ್ತು ಬಂದಂಗೆ ಆಗೋದು, ಆಯಾಸ, ಸುಸ್ತು, ವಾಮಿಟಿಂಗ್ ಆಗೋದು. Anemia, they will become completely weak. Hair loss, okay, loss of the hairs will be there. So where you have saw these symptoms in case of the cancer patients also, right? So that's why this is a deadly disease where it has to be avoided also. So with this, let's end up this, and we are moving on to the last part of the chapter. And that last part of the chapters we are going to study regarding the drugs and various different types of drugs and its effects along with the alcoholic effects. Okay, so let's end up our class. Thank you.